Okay, this is the first of 3 prep videos for PCR. Some of you have done 357 and or other courses and know perfectly well what PCR is, or perhaps have done research using PCR already, in which case you may find that this is not necessary for you. You might want to skim through the notes, and if they seem self-explanatory, you can probably skip the video. But for those that haven't, I want to provide this background because I think it is important when you're using a technique to have some good understanding of how it works, partly so that you can do controls properly, and partly just because it's helpful to know what's going on. Okay, so PCR is polymerase chain reaction, and it is a way of, out of an entire genome or a large sample of different types of DNA sequences, kind of picking out one particular sequence and making many copies of it. And that's the gist. There will be more. Just hang on. Okay, so we want to purify a gene for study. So suppose we are trying to amplify a gene so that we can transcribe and translate it in vitro and make a protein, and then test what the protein does. Maybe we want to do something forensic. We have a blood sample or something, and we would like to amplify, purify some parts of the genome so that we can identify the perpetrator. Maybe we are looking at how something segregates in a family, and we want to take samples of the same gene from different family members and try to make a connection between having a mutation in that gene and having the genetic condition. Okay, so those are all legitimate things that we might be trying to do. So we want to study this gene then, not just sort of segregation and pedigrees, but we want to know at the molecular level something about that gene. And we want to isolate just that gene sequence, possibly just from one individual with a disorder or from a bunch of family members, or maybe from an organism, so we can make a mutation in that gene and see what it does to the protein. Lots of different kinds of applications. So how do we do that? Now, if we think about proteins, proteins have a lot of distinctive features. You know, they have a particular charge. Some of them are hydrophobic, some are hydrophilic. They will have different combinations of the 20 different amino acids, some of which have been modified, maybe by methylation or phosphorylation or something. And it's quite easy to make an antibody that can pick out the specific features of a protein. And so we often use antibodies or other methods of separation based on the very different chemistry of different amino acids and different combinations of amino acids. The problem with DNA is that DNA all looks alike. There are no specific, charge differences from one gene to another, or the DNA is always positively charged. It's a sugar phosphate background with the backbone with the bases added, so it's not going to have different features like being hydrophobic or hydrophilic, or folding certain ways and things like that. So the DNA basically all looks the same, and chemically it's not easy to distinguish. So here's a long string of DNA just to kind of make that point. But if we can decide that we're trying to isolate, this gene here, it's possible that we can find some sequences that are unique. So what's really important here is that in order to do PCR, you have to know something about the gene. You have to know its sequence or the sequence of the DNA right to one side or the other of it. So we can maybe identify, small nucleotide sequences that are unique to that gene. That is sort of the key to PCR. So we don't actually isolate a gene away from others, but what we do is we can specifically copy many copies of just that sequence. And so, the idea of primers, which these lines are supposed to indicate, is the key part. Okay, so how do we find the gene? So the first problem is DNA all kind of looks the same. The sequences are heterogeneous. They're not exactly the same as each other, but chemically they don't behave that differently from one another. It is the actual sequence part that is the unique part of a particular gene. The second problem as well is that DNA is very tiny. So in order to study a gene, you're going to need, even to amplify with PCR and run some on a gel for diagnostic purposes, you're going to need millions of copies in order to do that. 1 or 2 copies, or 5 or 10 or 30 or 100, you're just not going to see on a gel. The detection is not yet sensitive enough for that. So Kary Mullis, this is him, who recently died, but he received a Nobel Prize. He shared it with, I think it was Michael Smith, who was at UBC at the time. And he invented this method, PCR. And PCR is a way of specifically amplifying DNA and specific pieces of DNA. So if you look at a chromosome, you have a way of, oops, that's not very good. You have a way of adding a little piece of DNA that matches one side and adding another piece of DNA that matches the other side. These are called primers. And then using a polymerase to make copies of the gene. And if you put these 2 pieces together, you'd have a double-stranded copy of your gene. Now, those of you who did 357 know that it's a little more complicated than this. There are sort of intermediate kind of linear products, but we're just going to look at the amplification of the intended product for this purpose, because we're talking about using PCR and not necessarily the detailed mechanism of how it works. So we needed the mind of Kary Mullis, and then we also needed something that came a bit later, was thermostable DNA polymerases. There are organisms that live in these hot pools and hydrothermal vents at the bottom of the ocean, where incredibly hot water is coming up, and these organisms are living at like 90 degrees. And that means that the normal temperature of most enzymes is hovering around 37 degrees. Think of your own body temperature. Your enzymes are going to work best at that temperature. But if you're living in this sort of seismically active place with these boiling pools, your enzymes are going to have to tolerate being at that very high temperature. So Kary Mullis, primers, and the idea of PCR, and then the thermostable polymerases is what made it sort of a doable process. So in the lab, towards the end of the semester, we're actually going to do PCR on dog DNA for a particular purpose, and we are going to be doing something kind of diagnostic, and you'll find out more about it, of course, when we get closer, because we're talking about the end of the semester. But you will actually be doing some PCR yourself. So what do we need if we're going to try to build a lot of pieces of DNA? You need a DNA template. Template is the word for the DNA sequence that is going to be copied. And it's going to be copied by, if this is the DNA and this is the primer, new nucleotides are going to be added one by one that are complementary to the template. So when we say we're copying the template, the reason why we're copying it is because DNA is double-stranded. I'll talk about that more in a minute. And so we're copying each of the strands. Now, you need the building blocks to make DNA. The first one is the primers. The primers are specific for the sequence that we're targeting, so they are part of the mechanism that specifies exactly what DNA we're going to amplify. But also, the DNA primers have the new nucleotides added onto them. So they form part of the product, and that's really important. So we need our building blocks. We, of course, need the right buffers, salt concentration, the correct pH, and so on, so that the enzyme will work. And then, in order to make it practical, we need a polymerase. The one we will use is TAC, but there are others that have different kinds of characteristics. You need a thermo-tolerant DNA polymerase. The reason is you're going to do cycles of heating the DNA to make it single-stranded, annealing the primers, so lowering the temperatures the primers can bind, and then extend. And you're going to do this, about between usually 25 to about 35 cycles. And so if you keep heating the DNA and then letting it cool, heating it, letting it cool, if you don't have this kind of polymerase, then you're going to kill the enzyme every cycle. And the first attempts at PCR actually required that you move the DNA from, this temperature water bath to another one, and then add more enzyme, and then back to the different baths and so on. And so it was labor intensive, and it was really expensive, because DNA polymerases, they're not super expensive, but having to put tons and tons of them into the reaction made it not very practical. Once we had machines that could increase and decrease the temperature, this is just a water bath with a rapid change of temperature. Having you put 1 microliter of your enzyme in there, and it can work. It doesn't get killed by increasing the temperature. We'll go through the cycles in a minute. So what kind of templates? Genomic DNA means all the DNA from the cells of the organism. And so it doesn't, and whatever tissue suppose you were grinding up mouse liver or something like that, you would have all the DNA for all the genes in there. And that includes sequencing between the genes. cDNAs are copies of messenger RNA. So that's just the sequences that are being expressed or transcribed in a certain stage, a certain tissue, whatever. And then if we have actually cloned a gene into a plasmid, this is a circular piece of DNA from a bacteria. We might have already engineered something that has the gene that we're interested in, but maybe we want to use PCR to make many copies of it and perhaps introduce a change into the gene, something like that. Okay, so the thermostable enzymes, as I said, they're isolated from organisms that live in thermal vents in the ocean or the hot pools in places like Yellowstone Park and some of these hot springs. And so the enzymes don't break down when you heat them. That's what thermostable means. There are slow enzymes that proofread, and if, for example, we wanted to actually express our protein, we would definitely want that kind of enzyme. Doesn't matter if it's a bit slow, although they're not bad. We want to make sure that sequence is correct. And so we would only have to maybe sequence a few of them to find one that we know is correct. When we do our dog project, we're going to use an enzyme with a proofreading function because we don't want errors in our product. Some are really fast and they have a higher rate of error, so the sequence isn't going to be perfect. But if we're just using that for diagnostic, if we're just using that to say, is there bacterial contamination in this food? Yes or no. This is supposed to be an arrow. Then the exact sequence isn't as important. We just want to see if we get a product or not. Okay, so there's different levels of accuracy, and it depends on what we're trying to do, what accuracy is acceptable. This is just a pretty picture of the kinds of places that these organisms live. This is like a hot pool, a natural hot pool. These are the hydrothermal vents in the bottom of the ocean. Okay, so I want to talk about the primers, and I want to talk about them, I hope, clearly enough without overwhelming, because this isn't primarily molecular course, but you do have to understand this. So when you get a file of DNA from a database or something, they will only give you one strand, and it will be the coding strand, if it's a gene that has a product. Now DNA is double-stranded, so that means the other strand is present, and it's going to be 5' to 3', you know, C, T, A, etc., T, etc. So it's going to be, it's going to be the reverse complement of the DNA. So when they give you the one, you already know what the other one is going to be. It's going to be the complementary sequence with the opposite polarity. Okay. Now we have two primers. We have a primer at one end of the pieces of DNA we want to amplify, and we have another primer at the other end of the piece we want to amplify. These are the actual primer sequences. And you will notice that one of them, called the forward primer, has the exact same sequence as the strand, the DNA sequence you've been given. The reverse is always the reverse complement of the corresponding sequence. So it's the same sequence as the bottom strand. And the reason for that is that the forward primer will recognize and bind to the bottom strand, and it will, the DNA will be made that's complementary. So it's making another one that looks like the top strand. And then the reverse primer actually binds and recognizes the top strand, and it makes complementary DNA, which is the same as the bottom strand. And that way you get both of the strands being produced. I think I have an image coming up. Not yet, though. So the PCR primers have to be complementary to the DNA sequence you're studying. So the forward one has to be complementary to the bottom strand. The reverse has to be complementary to the top strand. You want it to be unique to the DNA or the gene that you're studying. So there are sequences that are found repetitively, and they're found all scattered around the genome. You don't want that. You want something that is very specific to the particular gene. So your primers can only find and bind properly to one site. Okay. They need to have similar properties. They need to have the same annealing temperature within a couple of degrees, which usually means around the same GC content and so on. The idea is there is an annealing temperature that allows for your primer to find and bind to your DNA with maximum accuracy. So it will only bind to the correct sequence and not to one that's similar but has a few places where the nucleotides don't match. And so this is really important for making sure that the primers are only binding the intended target. And if you have one primer that is, has a lower annealing temperature and the other one has a higher annealing temperature, if you use the low one, the other primer can bind the wrong sequences. So this might be a little bit confusing. We can talk about it in lab if necessary. Okay. And so basically the idea here is that the polymerase allows you to go through many, many repeats, many cycles of amplification. And we continue until the nucleotides or the primers run out. So these are the cycles. There's a denaturing cycle, usually like between 10 and 30 seconds. The annealing, this is the temperature is lowered down to allow the primers to bind the DNA. And then extension, the temperature is raised to one of these two temperatures depending on the enzyme to allow the strands to be completed. And then, I would say most common 25 to about 35 cycles. I have seen PCRs that use more and fewer for different purposes, but on average we repeat this multiple times, and it's just in a little machine, a thermocycler that makes those temperature changes for you once you've put everything you need into the tube. Okay, so this is just a quick look. So here's a double-stranded DNA. If you were looking at this in a file, you'd only have the top strand, but I'm showing both so that hopefully it will be clearer what's going on. So at the denaturing stage around 95 or higher, the DNA becomes single-stranded. We lower the temperature down, and when it reaches the right annealing temperature, then the primers can locate and bind to their target sequences. These primers are a little bit shorter than normal, but it shows the idea of what's going on. And then finally, the sequences are extended. So this is where the enzyme is adding the right nucleotides until it gets to the end of the molecule. And so we end up now with two double-stranded molecules that both have the same sequence. And if you repeat this, about 30 times, you're talking about, I can't remember, 68 billion or something pieces of DNA. I'm trying to remember what the exact number is. But lots of them. And so this is our overall schematic. There's, you start with your template, you denature for, a few seconds, then the primers anneal, and then you extend, and so now you've got two, and then you repeat as necessary. Sometimes these commercial sites, I'm not advertising for them, They're selling products, but they also sometimes have very good explanations that are pretty clear about how the procedure works. Oh, and here it is, 68 billion copies, right? So if you theoretically, if you did 36 cycles and you went through and everything worked perfectly, you could end up with massive amounts. And this is just the idea. The sequence you want to amplify, you know, your primers are going to bind to these two positions, and then you're going to get all these pieces, right? Okay, and so the result of the amplification is that the piece of DNA between the two primers and including the primers is amplified exponentially. It slightly more than doubles, at least up to a point. And then the amount of product is related to primers and nucleotides. So once those are used up, you can't make any more product, which means we generally, when we set up a reaction, we use a few times more than we think we're going to need. So we put in excess amounts of the primers and of the nucleotides. And then only a very small amount of template is required. In theory, a single strand would be all that was required. And this is very important regarding things like forensic analysis, where sometimes you have very, very tiny samples, and yet you still, you know, that's all that you've got. So let's see how, you know, if we can identify the perpetrator or identify the victim, or, you know, whatever it is you're trying to do forensically. It theoretically will work well even with very tiny samples. Okay, and then for the next one, I will talk briefly about what we, you know, different uses of PCR, which you can skip if you need to for time, but I would maybe look at just through the slides and see that there's something related to DMAP1 in here that might be of interest. And then finally, just the use of controls, which are very, very important.